Hello, my name is Valencia Koomson, and I am a Dr. Martin Luther King Jr. visiting professor in the Department of Electrical Engineering and Computer Science at MIT, as well as associate professor in the Department of Electrical and Computer Engineering at Tufts University. Today, I will present the development of the HemoSensis device, a non-invasive point-of-care device for absolute tissue and cerebral oximetry. I will start my talk with a brief overview of near-infrared spectroscopy techniques to study light propagation in human tissue and its use in several applications. I will also review two widely used NIRS sensing modalities and NIRS applications in functional brain studies, optical mammography, and pediatric neurology. I will present the HemoSensis prototype device and system design and the core processing component, which is an application-specific IC that is used to give very high precision measurement of radio frequency, signal amplitudes, and phase changes. I will also present plans for a human validation study of the device at the MIT Clinical Research Center, along with preliminary results from lab experiments. I will end the talk with a few concluding remarks. The history of NIRS can be traced back to 1977, when Professor Jobs pioneered hemoglobin monitoring in the near-infrared range. The protein hemoglobin is a protein in red blood cells that carries oxygen. Hemoglobin collects oxygen from the lungs and delivers oxygen to cells throughout our body. The oxygen-loaded form of hemoglobin is referred to as oxyhemoglobin, while the one without oxygen is called deoxyhemoglobin. Almost all of our body functions are related to oxygen, so measuring the concentration of oxy and deoxygenated hemoglobin on a routine basis is desired in both academic and clinical fields. Although a system implementing NIRS techniques can be very considerably depending on the application, they're all centered around a basic principle of observing light interaction within the optical window in the near-infrared range. We can see that the absorption coefficients of oxygenated and deoxygenated hemoglobin differs significantly in what we call the optical window, this area highlighted in yellow, while the absorption from water is much weaker in this region. As a result, near-infrared signals can reach deep within the tissue. The concentration of oxygenated and deoxygenated hemoglobin can be extracted for measurement at different wavelengths. We are all familiar with the pulse oximeter that's used in clinical settings to measure heart rate and arterial oxygen saturation. Well, pulse oximeters are based on the continuous wave near-infrared spectroscopy technique, and it's the most classical method that's used. And it's benefited over 4 decades of development and refinement in theory and hardware for this type of device. In continuous wave NIRS measurements, low frequency or DC near-infrared optical signals are applied to the tissue, and we measure the amount of absorption of biological chromophores. But scattering losses cannot be measured directly in this method. Relative changes of hemoglobin concentration can be extracted by applying a differential form of what we call the modified Beer-Lambert's law. The most significant advantages of continuous wave NIRS are the simple hardware and low cost and compact design. Continuous wave NIRS is widely used in a number of consumer applications for fitness applications, sports medicine, and other types of applications. Here I show also the frequency domain near-infrared spectroscopy methodology. Here we present a multi-distance measurement where the optical signal is modulated at a high frequency radio signal frequency in the 80 to 100 megahertz range. And the modulation frequency is fixed, but we measure the degree of absorption and scattering at multiple distances away from the incident light. The modulated signals are applied to tissue at distances at several centimeters away. Here we can by extracting both the changes in amplitude and phase at multiple distances, we can look at the slope of the curves in terms of the change in amplitude and phase, and we can use that data to compute the absorption and scattering parameters associated with the tissue. From the measurement of the absorption and scattering parameters, we can apply the diffusion theory to actually extract the absolute value of the concentration of oxygenated and deoxygenated hemoglobin in tissue. Frequency domain near-infrared imaging techniques are opening up new pathways for in vivo optical characterization of biological structures. In studies where we're trying to understand the spatial temporal variations in absorption and scattering in diffuse biological media, deriving blood oxygen saturation levels and organelle concentration, and computing absolute oxy and deoxygenated hemoglobin concentrations. Here's an example of a neonatal study using NIRS. Here in a NICU environment and in a neonatal ICU, babies receive numerous interventions that impact oxygen delivery to the brain. But however, there's no direct measure of brain oxygenation within the incubator at the bedside. And our HemoSensis device is seeking to solve that problem. We have several applications of NIRS, one in functional brain studies to measure cognitive workload, for example, to study neurological disorders. It can be combined with EEG to look at to investigate epilepsy. Optical mammography applications where we look at changes in oxygenated tissue in breast tissue to search for malignant tumors. And as I just mentioned, neonatal infant monitoring, where we might be able to measure cerebral hemodynamics using near-infrared spectroscopy techniques, and also maybe biomarkers for autism have also been studied using these techniques. Current oximeters are very bulky and expensive. They use bulky, disparate, and tethered components. They measure tissue oxygen saturation only, and measurements of absolute concentrations of these biological chromophores can be very expensive. And also, it's not very useful for ambulating users, as you can see in the two applications shown in this figure here, where we have subjects being studied with large tethered devices that really hinder mobility. The future promise of NIRS lies in the ability to design miniaturized sensors. And that is the focus of our research, is in designing ASICs that combine a number of different functions, both advanced analog signal processing, digitization, all on a single piece of silicon. Here we have one generation of our ASIC design in a 180 nanometer CMOS process, where we've combined all of the functionality needed for amplitude and phase extraction of the optical signal. We have a programmable gain function to increase dynamic range. We use ultra low noise bipolar front-end amplifier devices, bipolar devices and front-end amplifiers to be able to detect very weak optical signals. We use a dual channel measurement for high resolution phase measurement, and we have on-chip demodulation to minimize noise. We have carried out both characterization of our ASIC designs using both solid and liquid tissue phantoms. For FD NIRS, for the frequency domain NIRS, characterization of the system level error and is quantified using both the solid tissue phantom with a calibrated absorption and scattering parameters, and a liquid tissue phantom that's composed of a milk, water, and India ink, which mimics the absorption of oxygenated and deoxygenated hemoglobin. Characterization results show results that are comparable to a commercial oximeter. So here we show our prototype device, and we look at the data distribution in terms of the amplitude and phase measurements. We look at the amplitude uncertainty, phase uncertainty, and the uncertainty and absorption and scattering parameters due to system level error analysis. And we see a direct comparison here in our work here. We get less than 0.5% in terms of amplitude uncertainty and 0.5 degree uncertainty in phase measurement. The miniaturized system for the HemoSensis device, we built a low-cost prototype device for human validation studies. Here it shows the sensor probe is a passive probe that's fiber coupled to a multi-layer printed circuit board, where we combine our custom designed ASIC, a high gain avalanche photodetector, and a time multiplex laser array that's integrated on the PCB. And the control, calibration, signal processing are all realized in a graphical user interface in LabVIEW and MATLAB environment. Here at our human validation study protocol, we have two different tests that we're seeking to do to validate the system. One is a venous occlusion test and an arterial occlusion test. And so we place the sensor probe onto the forearm and we apply a blood pressure cuff near the elbow. For venous occlusion, we have the subjects at rest for 30 seconds, and then we apply a pressure, a 60 millimeter mercury pressure measurement is applied to the subject, and then we release that pressure and perform measurement for 30 seconds. For arterial occlusion, we apply much higher pressure in order to block both arterial and venous flow. Right. For a breath holding test, we calibrate the device using a solid phantom. We position the probe on the forehead. We have the subject at rest for 30 seconds. We ask the subject to hold the breath for 30 seconds, and then we have another 30 seconds of rest. Here is some preliminary results using our protocol for venous occlusion test results. Here we've identified a specific muscle on the forearm where we've placed the probe. We placed the arm at heart level. And you see here, we have the patient at rest for 30 seconds. We measure both our system. These are results of our device measuring both oxygenated and deoxygenated hemoglobin. At the 60-second mark, we apply the pressure cuff for another 60 seconds, and we see an overall increase in the oxygenated blood flow as expected. And this shows the total hemoglobin concentration increase during the experiment and a computation of the hemoglobin oxygen saturation. Okay. So in conclusion, near-infrared spectroscopy techniques are creating pathways towards new applications to study biological tissue, both in functional brain imaging, cerebral oximetry, stroke assessment, and optical mammography. Silicon IC technology provides a small footprint and low-cost, low-power platform for both continuous wave and the frequency domain near-infrared spectroscopy applications. And the computational power of silicon technology can address the engineering challenges of developing a robust and portable FD-NIRS probe for both research and clinical studies. Thank you.